remarks, Dr. Asher. Next is Dr. Stephen Quay. He's a physician and a scientist with hundreds of published scientific articles. Dr. Quay has been cited over 10,000 times, holds 87 patents across 22 different fields of medicine, and he's invented 7 different FDA-approved medicines. Further, he conducted an early analysis of COVID-19, which has been downloaded nearly 200,000 times. He's an expert in the field and brings an upper echelon of analysis to the question we're posing today about the origins. Now to you, Dr. Quay.
Thank you, Congressman Scalise and Comer, members of the subcommittee of the coronavirus crisis, ladies and gentlemen. I appreciate the nonpartisan approach the subcommittee is taking today. Clearly, science in the last few years, but especially on the topics related to the COVID pandemic, has been co-opted by geopolitics. Thus, I am here not as a mouthpiece for any particular party, but as an American scientist. I dedicate my testimony today to the more than 600,000 Americans that this pandemic has killed so far, with the hope that by clarifying the origin of COVID, we can help to prevent future pandemics and the loss of innocent lives. Given this backdrop, I will be keeping my remarks today to matters of science. I'll state facts and evidence about the pandemic that I have collected. I'll explain why those facts lead to certain conclusions, allow a particular inference, or rule out a hypothesis about the origin. Today's testimony is an effort to answer the question posed on December 30th, 2019 by Dr. Zhengji Shi, the head of coronavirus research at the Wuhan Institute of Virology. She had just been told that a novel coronavirus had caused an outbreak of pneumonia at hospitals close to her laboratory in Wuhan. In what I believe was her last unscripted public utterance, she has been quoted as saying, "Could they have come from our lab?" In the last 18 months, we've learned an intense amount about the origin of the pandemic, but one of my frustrations is that virologists and science writers around the world seem to want to ignore what has been learned and the inevitable conclusion it reveals. As inconvenient as it is, I believe the evidence conclusively establishes that the COVID pandemic was not a natural process, but instead came from a laboratory in Wuhan, China, and that it has the fingerprints of genetic manipulation through a process called gain-of-function research. There are 6 undisputed facts that support this hypothesis. Now, as a reminder, the top left there, a little science. As a reminder, all zoonoses are diseases that involve a virus infecting an animal, the animal coming in contact with a human, and then the human being infected. The question of the origin of a zoonosis is the question of the location of the animal. In natural zoonoses, the animal is in nature, a cave, a farm, or the market. The infected human comes in contact with the animal and gets sick. For lab-acquired zoonoses, the animal is in a laboratory or in cells from an animal in a petri dish, and the human works in that lab. In deciding between a natural origin and the lab origin, there are 6 facts no one disagrees with. Number 1, the Chinese first told the world that the COVID pandemic began in the Hunan seafood market because approximately 1/2 of the early cases had been associated with that location. This would have been reminiscent of the 2 previous coronavirus epidemics, as both SARS-1 and MERS began in live animal markets. However, after 18 months of investigation, we now know it did not begin in a market in Wuhan for the following 3 reasons. First, none of the 11 patients from the Hunan market or any other market in Wuhan were infected with the earliest virus, meaning they came into the market already infected. The 4 patients with the earliest genetic version of the virus all had one thing in common. None had any exposure to the Hunan market. Second, we know in January 2020 from public scientific databases that the patient with the earliest first complete virus sequence was a 39-year-old man treated at the General Hospital of Central Theater Command of the People's Liberation Army of China, located on Wuhou Road in the Wuchang district of Wuhan, about 3 kilometers from the Wuhan Institute of Virology. After 18 months and almost 3 million viruses sequenced worldwide, this virus still remains the earliest. Second, none of the environmental specimens from the market had the earliest virus, meaning they also came into the market. This included 1,055 samples of frozen food in the market, all of which tested negative for COVID. Third, 457 animals from Hunan market were tested. All were negative for COVID. 616 animals from suppliers to the market were tested. All were negative. 1,864 wild animals from southern China of the type found in the market were tested. They were also all negative. These data from the publicly available WHO report established that it did not begin in the Hunan market or any other market in Wuhan. Number two, failing to find it in any market in Wuhan, the search was expanded to all of China. But after testing 80,000 samples from 209 different species from other markets, farms, and nature, the virus has not been found in a single specimen. The probability of this for a community-acquired infection is about 1 in a million. This is what you'd expect for a lab-acquired infection. Number three, after testing 9,952 stored human blood specimens from hospitals in Wuhan from before December 29th, there was not a single case of COVID in any specimen. It was expected that between 100 and 400 would be positive. The probability of this for a community-acquired infection is also about 1 in a million. But this is what you'd expect for a lab-acquired infection. There is no evidence of multiple animal-to-human transmissions. With two prior coronavirus outbreaks, SARS-1 and MERS, 50 to 90% of early cases were from various animal-to-human infections. For SARS-CoV-2, 249 early cases of COVID-19 were examined genetically, and they all were human-to-human transmission. For a community-acquired infection, this is the probability of tossing a coin 249 times and getting heads every single time.
Wow.
This is, however, what you'd expect for a lab-acquired infection. Number five, SARS-CoV-2 has a unique trigger on its surface called a furin cleavage site and a unique code in the genes for that site called a CGG dimer. So these are two independent levels of uniqueness. The furin site is why the virus is so transmissible, why it invades the heart, the brain, and the blood vessels. Viruses like Ebola, HIV, Zika, yellow fever all use furin sites, and this is part of the reason for their deadliness. But the entire group of coronaviruses in which SARS-CoV-2 belongs does not contain a single example of a furin site or a single example of the CGG dimer code. A comprehensive study by MIT group shows that there has never been a furin site or the CGG letter dimer in this group of coronaviruses for at least 1,000 years, since William won the Battle of Hastings in 1066 to become monarch of England. But since 1992, in gain-of-function experiments, the WIV and other laboratories around the world have inserted furin sites into viruses repeatedly. It is the only sure method that always works and always makes them more infectious. And the CGG code found in COVID-19 is commonly used in laboratories around the world, including the WIV. You can literally order it from a supply company on the internet.
Bust it. Just bust the move.
By the way, some virologists have said, well, but Dr. Quay, furin sites and codon dimers occur in one or more other of the four classes of coronaviruses, and with a process called recombination, a COVID-like virus could pick one up. This is wrong for three reasons based on well-known fundamental biology, probably college level. Because coronaviruses do exchange genetic material so easily, the very existence and stability of these five distinct groups is evidence from nature that there must be barriers for exchange of genetic material. I've spoken previously about the 1,000-year stability of the group of COVID that COVID belongs to. If recombination was easy, this group would have merged into the other four into a single group. Two things stop recombination from happening. And this includes research by Dr. Dasikan Shi, by the way. First, recombination happens when one poor bat is infected with two different viruses, and during that co-infection, the genetic material gets exchanged. So here, the group of viruses that have furin sites and CGG dimers don't infect the same bat species as the COVID-like viruses that don't have furin sites. So the COVID viruses can only exchange genetic material within their group, the group that hasn't had a furin site or a CGG dimer for 1,000 years. Now, second, even if you force two different viruses into a cell in the laboratory, there are unique genetic sequences that are required for gene combination or recombination to occur. The hotspots where exchange can occur are different for each group and incompatible between the groups. In a clever use of this concept, Dr. Ralph Baric, a leading coronavirus scientist from North Carolina, once made a vaccine candidate that was resistant to recombination. He didn't want to recombine after he put it out in the world. He did this by using synthetic biology to change the hotspot gene sequences. Number 6, finally, SARS-CoV-2 was pre-adapted for human-to-human transmission from the very first patient. Specifically, the part of the virus that interacts with human cells was 99.5% optimized. When SARS-1 first jumped into humans, it had only 17% of the changes needed to cause an epidemic. As evidence of the relevance of this work, the UK strain, remember last fall, in the fall of 2020 that emerged and was more effective, was a change in one of those 17 few spots that hadn't been optimized at the beginning, N501Y. Some virologists may claim COVID virus was not pre-adapted and point to evidence it has been mutating a lot since it first emerged, making new variants like the Delta strain currently in the news. But the details tell a different story. COVID does randomly make, on average, about one genetic mistake every 2 weeks. So after a year of circulating the globe, it will naturally have, on average, 26 changes somewhere in its genetic code compared to when it started. But the vast majority of these mistakes are either neutral, having no effect on the deadliness of the virus, or actually detrimental, making the virus weaker. Less than 1% of the changes actually improve the virus. So how do I believe the COVID virus was taught to infect humans in a laboratory? A commonly used gain-of-function method to optimize the COVID virus would have been to serial passage in a laboratory on a humanized, genetically modified mouse that can develop a human-like pneumonia. You take 20 mice, you infect them, you wait a week, and then you recover the virus from the sickest mouse. Then you take another 20 and you do it again, and suddenly you're starting to kill the mice. And finally, after several weeks, through this directed evolution, you'll produce a mouse that can kill every humanized, a virus that can kill every humanized mouse. However, it's actually a challenge to create that humanized mice for the serial passage in the first place. Here, the WIV has acknowledged that for several years they've worked with humanized mice developed in Dr. Ralph Baric's laboratory in North Carolina and funded at U.S. taxpayers' expense. In closing, let me say to the families of those who have died from COVID, families of those who have died from COVID, I'm dedicated to applying my scientific and medical expertise to understanding how this pandemic happened, and importantly, helping put safeguards in place at our research institutions to be sure this never happens again. Thank you.
Riveting testimony, Dr. Quay. Appreciate the work you've done in sharing that. And now finally, to bring us to Dr. Richard Muller. Dr. Muller is a professor emeritus of physics at the University of California, Berkeley. He's worked across his field of study, leading the two efforts that have won Nobel Prizes and the National Science Foundation Alan T. Waterman Award for outstanding research. His scientific expertise has been vital to the work that we're doing. Dr. Muller, appreciate you coming.
Thank you very much, Mr. Comer, Mr. Scalise. I appreciate you doing this. It's extremely important. Having just listened to Dr. Quay's comments, I think we need to recognize the power of science. And previous speakers have said that we may never know the origin. I think that is incorrect. I think when we bring the power of science to this, we can reach conclusions that are very powerful. Dr. Quay has covered most of the science that is most relevant. I would like to emphasize 5 points, each of which is capable of separating and distinguishing between a natural origin, a zoonotic origin, and the lab origin. So let me just draw your attention to these 5. The first one is the absence of a pre-pandemic infections. He mentioned that. And with over 9,000 taken in Wuhan, no pre-pandemic infections. That's unprecedented. Didn't happen in MERS, didn't happen in SARS, doesn't happen with other viruses. Unprecedented. It's very difficult to explain. Papers are being written trying to explain it, but those papers aren't evidence. They're what you do when you're stuck and the evidence disagrees with you. The absence of a host animal. If you look at the famous Lancet article, one of the reasons they say you can dismiss these conspiracy theories of lab origin is because they explicitly say China has identified the host animal. They've identified the location, and they even praise China for its openness. This paper, The Lancet, does not read well when we look at it 16 months later. I recommend everybody reread it and see whether there's any value in that. The unprecedented genetic purity that Dr. Quay talked about. Again, MERS, SARS, previous viruses don't have this, but it is exactly what you would expect if you've gone through gain of function. So each one of these things is compelling by itself. If we had any one of the five things, these are three of them, we should conclude that the evidence strongly favors the lab origin. The spike mutation. The fact that there's no known way for that spike mutation to get there other than by gene insertion in a laboratory is a very powerful argument. And again, there are papers written that try to explain this away, but those papers aren't presenting evidence. The evidence in favor of the natural zoonotic origin, there isn't any. The only evidence that's cited is, well, it looks like another zoonotic case, or those cases are quite common, but there's no scientific evidence supporting those. And finally, the fact that this virus was optimized to attack humans. Again, something that has never happened in natural releases.
What is the one thing every Coldwell Banker agent has in common?
But it does happen if you run it through the gain of function. So there's really all of the scientific evidence argues in favor of the laboratory origin. The papers against it, the famous paper by Christian Andersen in Nature Medicine, his argument against the lab leak case was his claim that the spike protein had not been optimized, and it certainly would have been. That is his one argument against it in that paper that is so widely quoted. But recent evidence, as Dr. Quay talked about, shows that it was 99.5% optimized. I'm going to end with a little anecdote, a story of that is as horrifying and more frightening than almost anything else in my life. In the early days when I was trying to teach myself virology, I called several of my friends who are expert in virology. One of them, I asked him if he could help me review some of this literature, the literature that suggested that there was a lab leak. And he said, no, I'm sorry, Rich, I can't do that. Well, okay, is there someone in your lab who could do it? No, nobody in my lab will do it. Well, why not? Well, let me be candid. If anybody in my laboratory is discovered to be working on a laboratory leak hypothesis, China will label us enemies of China, and the laboratory will be blacklisted, and we will no longer be able to collaborate. We collaborate all the time with China. Nobody will take that risk. This one of the most chilling conversations I've had in my life. The idea that China has managed to interfere, to break the United States freedom of expression, freedom of investigation, freedom of thought by through this collaboration effort, really scary. So let me just say that some people say we will never know, not until China confesses. Or unless there's a whistleblower. Well, we have a whistleblower. It was the virus itself. It came here, it came out of China, it came to us, and it carried with us genetic information. And there are, in my mind, 5 compelling sets of scientific evidence that allow us to reach this very strong conclusion that yes, it was a laboratory. Thank you very much.
Well, thank you.